Hi viewers, this is J. Swami, Assistant Professor of Zoology. Today we are going to discuss the ATP synthesis. It is also known as oxidative phosphorylation. ATP synthesis takes place in the presence of an enzyme complex which is known as ATP synthase. This enzyme mediates the catalytic activity of conversion of adenosine diphosphate into adenosine triphosphate by adding an inorganic phosphate. Now, the ATP synthase, as we are seeing, this entire it is the ATP synthase enzyme. It is also known as the enzyme complex 5, multi-protein complex 5 in the electron transport system. Now, this ATP synthase enzyme is present in the inner membrane of the mitochondria. Now, let us see the structure of ATP synthase enzyme. Here, this is the structure of the ATP synthase where we find out two subunits. Otherwise, we can divide this into two parts. This is known as F0 or F0 and the above part is known as the F1. There are two active sites or functional sites. Otherwise, there are two functional sites in the ATP synthase. One is known as the F0, another one is known as the F1. So that the ATP synthase is also known as F1 F0 ATP synthase. F1 F0 ATP synthase. Now, see the structure of F1. Let us discuss the structure of F1. F1, it is a peripheral membrane protein. The F1 represents, this is the first factor of oxidative phosphorylation. The number 1 represents this is the first factor of oxidative phosphorylation. So that it is known as the F1, first factor of oxidative phosphorylation. And it was identified and purified by the Ephraim Racker. The scientist name is Ephraim Racker and his group. Ephraim Racker and his group identified the F1 factor and purified the F1 factor. And if you observe the ultrastructure of the F1, there are 9 subunits. Out of these 9, there belongs to 5 groups. Namely alpha, beta, gamma, delta, epsilon. Alpha, beta, gamma, delta, E groups of subunits are there, but they are totally 9. Thereby, alpha subunits are 3 alpha subunits, 3 beta subunits, 1 gamma subunit, 1 delta subunit, and 1 epsilon subunit. Totally 9 subunits are there, which are belongs to 5 groups. Out of 9, alpha 3, beta 3, gamma 1, delta 1 and epsilon 1. Let us here it is see. This is the alpha subunit. There are 3 alpha subunits, and the yellow colored one, they are the beta subunits of about 3 beta subunits are there. And here it is blue color one, gamma subunit, and here it is green color one, delta subunit, and here epsilon subunit. Totally 9 subunits which are belongs to 5 groups. Out of 9, alpha 3, beta 3, gamma, delta and epsilon, each one contribute 1 subunit. Out of these 9, the gamma subunit will be act like a column-like structure which forms a central axis, vertical central axis. It acts like a vertical axis which is present in the center. And around this vertical axis which is made by gamma, all the alpha and beta subunits, they are arranged alternatively. They are arranged one by one alternatively. 3 alpha subunits and 3 beta subunits, they are arranged alternatively upon the central axis of the gamma. Now, the specialty of beta subunits is each and every beta subunit. There are 3 beta subunits. Each beta subunit contain 1 catalytic site. So, there are 3 beta subunits. Out of 3 catalytic sites are there. What is a catalytic site? Where there is a conversion of ADP into ATP takes place. ADP into ATP takes place. That is the site where actually the synthesis of ATP going on. So there are 3 beta subunits. Each and every beta subunit should contain a catalytic site where actually the synthesis of ATP takes place. So this is all about the F1 portion. This is known as the F1 active site. Then coming to the F0, F0 portion. The O or 0 represents the sensitivity, indicator for the sensitivity of oligomycin. Sensitivity of oligomycin. This F0 portion is a integral membrane protein. Now, this integral membrane protein acts like a proton channel. It acts like a proton channel. F0 is an integral membrane protein, and which acts like the proton channel. And in the F0, unlike the F1, we have subunit A, subunit B and subunit C. A, B, C subunits are there. In the F1, as we know, alpha, beta, gamma, delta and epsilon are there. So here in the F0, we have only 3 kinds of subunits: A, B and C. And if you see the ratio, A 1, B almost all 2, but whereas the C, it may be 10 to 12. Here we will see the number also. Here A, only 1 subunit is there. A, only 1 subunit we are seeing, and B, there are 2 subunits. But here around the C, here 1, 2, 3, 4, 5, 6, 7, around that there are 10 to 12 subunits are there. 1 is to 2 is to 10 to 12. The representation of A is to B is to C. So they are arranged in such a ratio. And now, if you observe the ATP synthesis process, where the beta subunits of the F1, they have the active sites upon their surface, catalytic site, where actually the ADP plus inorganic phosphate, they are converted into ATP plus H2O. So it is a reversible reaction. ADP plus Pi gives us a formation of ATP and H2O. Although there is a formation of ATP upon the catalytic site of the beta subunit, as there is a synthesis of ATP on the catalytic site of beta subunit, but the synthesized ATP molecules may not be released from the surface of the beta subunits. Otherwise, may not be released from the ATP synthase enzyme. So that in order to relieve that, there will be a establishment of the proton gradient. There will be a establishment of the proton gradient on either sides of the membrane. On either sides of the membrane. Right? Now, during this time, whenever the ATP synthesized and get attached to the surface of the ATP synthase, now there will be a movement of protons from transmembrane region to the matrix of the mitochondria. Transmembrane region to the matrix. There is a flow of protons. Otherwise, movement of protons through proton canal. Proton canal which is made up of F0. Right? Now, there is a movement of protons from transmembrane space to matrix through the proton channel of the F0, which is an integral part of the ATP synthase enzyme. Now, due to this, there will be establishment of the proton gradient on either sides of the membrane that tend to release the ATP from the surface of the ATP synthase enzyme. At the same time, due to the flow of the protons through the proton channel of the F0, the column of the C subunits, the column which is made of the C subunits, and as well as the vertical axis which is made up of the gamma subunit, okay, they rotate perpendicular to the membrane. They rotate perpendicular means like this. They rotate perpendicular to the membrane, and thereby here we have seen the rotation axis also like this. They rotate perpendicular to the membrane, and thereby the catalytic sites of the F1, which are represented by the beta subunits, they are involved in the catalytic sites of the beta subunits of the F1 portion. They one by one alternatively involve in the ATP synthesis. They are involving in the ATP synthesis one by one. So this is known as the rotational catalysis. Rotational catalysis. This is known as the rotational catalysis. That is, due to the perpendicular rotation of the gamma subunit of the F1, the column which is made up of C subunits of the F0, they rotate perpendicular to the membrane, and thereby the catalytic sites of the F1, they involve in the protein synthesis one by one alternatively. Such a kind of phenomena is known as rotational catalysis. This theory was proposed by Paul Boyer. Paul Boyer proposed the rotational catalysis theory. This is all about the synthesis of the ATP. This is all about the synthesis of ATP. See the terms. Okay, all of them have been covered. Then move to the next slide. So F0, ABC subunits and their ratio. Okay, the reversible reaction and role of the F0, rotational catalysis. Okay. So this picture is somewhat clear here. We will see all the subunits, 3 alpha and 3 beta. So light blue color is alpha, and somewhat thicken, they are the beta. So here it is gamma, delta and epsilon. So C, A, B, like this. Then come to the next process, energy budget of cellular respiration and the discrepancy in the number of ATP molecules. What is the energy budget is, if you take into consideration the oxidation of one glucose molecule in the cellular respiration, how many number of ATPs are utilized and how many number of ATPs are being gained. How many are spent and how many have been gained in the cellular respiration is nothing but the energy budget. That is the number of ATPs utilized, number of ATP molecules utilized in the cellular respiration, and the number of ATP molecules which have been derived from the, produced from the cellular respiration is nothing but the energy budget of the cellular respiration. And there is a discrepancy in the number of ATP molecules too. Why? Because if you observe the electron transport system, now the ATPs may be formed from the NADH or FADH, and the number of ATPs which have been formed from the, produced from the either the NADH or FADH, they solely depend upon the consumption of oxygen, absorption of oxygen by the mitochondria for the oxidation of NADH to NAD and FADH2 to FAD plus. So that is the consumption of oxygen by the mitochondria for the oxidation of NADH and FADH2, that dictates, that determines the number of ATPs which have been going to form in the cellular respiration. This is known as the P by O ratio. P by O ratio. So here the P represents the phosphorylation and O represents the oxygen consumed, the consumed amount of the oxygen. Now, if the electron donor is NADH, let us assume if the electron donor is FADH, NADH, then the P by O ratio is, ATP to half of O2 ratio is 2 to 3. If the electron donor is NADH. Otherwise, if the electron donor is FADH2, the P by O ratio may be 1 to 2. 1 to 2. That means that if the electron donor is NADH, 2 or 3 ATPs going to form, and if the electron donor is FADH2, 1 or 2 ATPs being formed from their substrates. Right? So, what the scientists thought that the ratio should be constant, the P by O ratio should be constant, so that they set to a constant point. They set the P by O ratio to a constant point. That is, the P by O ratio for the NADH, that is 3 instead of 2, and the ratio P by O ratio for FADH2, it is 2. That is, whenever the NADH is a electron donor, the ATPs may be formed. If the electron donor is FADH2, then that time 2 ATPs going to form. So they have stabilized the amount. They made it constant. So here this calculation have been in practice for years together. This P by O ratio, that is for NADH it is 3 and FADH it is 2. It has been in the practice for years together. But according to the chemiosmosis model, one pair of electrons, if they are transported to NADH, according to the chemiosmosis model or theory, if 2 electrons are transported to NADH. It requires, otherwise it pumps 10 protons. It transports 10 protons from mitochondrial matrix to transmembrane space through the NADH dehydrogenase complex. If 2 electrons are transported to NADH, 10 protons being pumped from mitochondrial matrix to transmembrane space. And that indicates that in order to synthesize one ATP, the NADH requires 4 protons per each ATP, so that it becomes 2.5. So previously we thought that from 1,1-ADH there are 3 ATPs may going to form, but according to the chemiosmosis theory, so it become 2.5 instead of 3. Now if the FADH2 is the electron donor, 6 protons being pumped from matrix to the transmembrane space, right? Then thereby for 1 ATP 4 protons, so that it is 6 by 4, it becomes 1.5. It becomes 1.5 instead of previous 2. So according to the chemiosmosis, whenever the NADH is the electron donor, 2.5 ATPs have been formed. If it is FADH2 is the electron donor, then that time 1.5 ATPs has been formed. This is the new calculation. And one more thing, the pair of electrons which are released from the glycolysis, in order to reach the electron transport system of mitochondria, they use different kinds of shuttle systems in the different cells. For example, if they are transported to FAD, they use glycerol phosphate shuttle. If they are transported to if they are given to FAD, then they use glycerol phosphate shuttle. If at all they have been given to NAD, they use malate aspartate shuttle. For example, in the brain, the electrons are transported to FAD. Okay, that is, they use the glycerol phosphate shuttle, and the amount of ATPs are produced for FADH2 is 1.5 ATPs. If the same thing, if it happens with the NAD in the cells like the cardiac muscle cells and the hepatocytes of the liver, then during that time on NAD or NADH is equal to, that is, 2.5 ATPs. So let us see the discrepancy in the number of ATP molecules in between the old calculation, previous calculation, and the new calculation. Now here we are seeing the reaction product ATP yield in the previous present ATP yield according to the chemiosmosis theory. Now here let us see, in the glycolysis, as we know, there is a release of dehydrogenation of reaction. There will be a conversion of NAD into NADH. So we have 2 NADH and we have 2 ATPs in the glycolysis. So according to the previous calculation, 2 NADH is equal to 6. 2 into 3. 2 3 is 6. 2 ATP means only 2 ATPs. Okay, but here it is, we are observing the 2 into 2.5. 2 into 2.5 is equal to 5. So that is, there is a calculation difference. Here it is 6, but here it is 5. If we come to the pyruvate oxidation, 2 per glucose means 2 NADH. Previously it gives 6, but now it is only 5. If you come to the oxidation of acetyl coenzyme A, 2 for each glucose molecule, 6 NADH, 2 FADH2 in the Krebs cycle. 6 3 is 18, but it is become 15. 6.25 is a 15. 2 FADH2 is equal to 4, but here it is 3. So 1 ATP get converted to, 1 GTP get converted to 1 ATP, so that total 2 GTP is equal to 2 ATPs. So there is no change in the calculation of the GTPs. So totally here there will be a previously from one glucose molecule we may yield 38 ATPs, but here it is only 30 because of the discrepancy in the number of ATP molecules that have been released. So this is about the difference in the ATP molecules. So if you observe the chemiosmosis in some of the animals, chemiosmosis takes place in all the organisms. Either they may be the prokaryotes or eukaryotes, plant cells, animal cells and all. Now apart from the prokaryotes, the chemiosmosis takes place near the plasma membrane, whereas in the eukaryotes, that is in the cristae of the mitochondria, chemiosmosis takes place in the cristae of the mitochondria, where in the case of plant cells, that takes place, chemiosmosis takes place in the thylakoid membrane of the chloroplast, right? And finally what we know is, in the chemiosmosis process, in order to synthesize one ATP, the ATP synthase enzyme that requires 12 kilocalories of energy. It requires 12 kilocalories of energy for the synthesis of one ATP molecule, otherwise 50 kilojoules. It requires 50 kilojoules of energy, right? And if you see the brown fat chemiosmosis, the chemiosmosis of the brown fat, very useful fat, right? So that is in the infants and as well as the young mammals, they have the brown fat. It plays an important role in the regulation of the body temperature. It may be located in the dorsal region above the vertebral column or in the neck region. So in this brown fat we will see enormous number of mitochondria and as well as many number of cytochromes, so that it looks like in brown color. And the tissue which is present in this, that releases more and more amount of heat, and that responsible for the maintenance of the body temperature at the 37 degrees Celsius. And this heat may be released from the inner mitochondrial membrane. There will be a proton motive force. From this the energy will be released, right? And the brown fat, that is responsible for the maintenance of the regulation of the body temperature. This is all about the ATP synthesis, otherwise oxidative phosphorylation. So under this we have ATP synthase enzyme and its parts and their responsibilities, and as well as the discrepancy in the number of the ATP molecules whenever we compare to the previous calculation and the present calculation. Thank you one and all.